Hello, everyone. I would like to welcome all of you in my PhD defense. So today I'm going to talk about deep generative models for supporting prostate cancer diagnosis on MR images. And the outline of today's presentation will be an introduction where I will explain prostate cancer, MRI images, CAD system, GANs, as we had it in my trial lecture, and then the aim of this study, thesis, a summary of the paper, discussion, and a conclusion. So to begin with, prostate sits deep in the pelvis, below the neck of the bladder and close to the rectum. The prostate is a gland of the male reproductive system, and the prostate produces prostatic fluid that enhances semen health and mobility. Prostate cancer in 2020 was the second most common type of cancer in men, the second in terms of incidence in men, and the fifth most common cause of death in men. Specifically, in Norway, PCA was the most common cancer in men between 2019 to 2023. Now, patients can have routine checks or some symptoms. So if a patient wants to check, it goes for a blood examination, and then they have this PCA, prostate-specific antigen, where any elevations can be an indication there might be cancer, and also they might have digital rectal examination by the doctor, the radiologist. And if there are indications, then maybe that patient will go for an MRI scan. Now, MRI resonance image has shown tremendous potential due to its non-invasive nature. It takes advantage of the nature magnetic properties of the human body. And with MRI, we can produce images of the anatomy and the functional process of the body. Now, specifically, when we have a suspected PCA, there is the multiparametric MRI protocol recommended as an initial diagnosis for that. And we have the T2 weighted sequences that provides structural and anatomical information. We have the diffusion sequence that quantifies the extent to which water molecules move in the tissue. And we have two derivatives here, the high B values and the ADC. And lastly, we have the DCE, dynamic constant enhanced images, where we use them to study the vascularity characteristics of the tissue. Now, with DCE, we have to inject to the patient gadolinium, and there are current controversials on the usage. So there is the usage without the DCE is called Biparametric and has shown similar results with the Biparametric. And in this study, we're using Biparametric. Now I would like to show you how the cancer looks like. So here in the green box, we have the prostate area. And then the cancer in the T2 and ADC appears as a darker spot, whereas in the high B values it's a bit brighter, it's brighter. So this might seem easy, but for a doctor it's difficult to assess it. And therefore, when we do MRI interpretation, we have the PI-RADS, Prostate Imaging Reporting and Data System, where we can, the radiologist go and checks the probability of PCA presence. And it scales from low to high. However, there are challenges because doctors might have different opinions. So we have the challenge of inter-observer variability. We have time requirement to do so. And we have also problems with scalability because time requires for that. Overall, the detection of PCA remains a challenge even if we combine with Biparametric in combination with PI-RADS. So now, if we have the MRI and MRI examination has shown that maybe there are potentials for cancer, then the radiologist will send this patient for a biopsy. So a sample will be taken, and then there will be a histopathological evaluation, where means the doctor will assess the aggressiveness of the cancer. Overall, we need to have accurate diagnosis, which is important to avoid over and under treatment. And this is not important only for the last step, but it's important here, because we don't want to patients that they don't have cancer to go for a biopsy. So when a doctor has the biopsy, needs to assess it. So there is a 5-grade classification system called ISUP, International Society of Urological Pathology. And here we separate patients with indolent PCA and patients that might have lethal or they might die from this cancer. Now, we have computer-aided detection systems that can help us in many of the stages of this pipeline. So those systems can help with qualitative analysis. They can be faster. They can reduce the inter-observer variability. And overall, they provide a standard way of image interpretation. Those tools have used in medical imaging to support radiologists in making important decisions, and also have been used in PCA, in the context of PCA. So how those tools look, CAD systems, those systems, is like we give those systems an image, and then those can output if there is cancer or not. So we have probability maps, can have detection maps, the number of lesions, the location, and maybe the PI-RADS score. So let's see the overall pipeline of this system. First, we have the data acquisition. We acquire the images. The second step is pre-processing. So here we do some denoising, MRI bias field correction, and normalization. Another very, very important step is the cropping of acquiring the prostate areas of those images that then we can use for training the models and the different stages. We can also use that when we apply it in clinic. So, the next step is the segmentation, where we segment automatically the prostate area. After that, we do registration. We bring all the images in the same size. And then we have trained the deep learning model that then we provide those images, and then that model outputs can be detection maps or hotspots, so the indication or where the cancer is. And in the last step, we have the diagnosis, where we give this information to the doctor, and then the doctor can send that patient in a biopsy or not. Now, the current CAD systems for PCA, they have been successfully implemented. Common architectures are UNet, and those models use fully supervised learning, which means they need these pixel-wise annotations for training, as you can see on the left side with the red area, which is again time-consuming and prone to potential biases. And also because this takes time, might be challenges with scalability. So generative adversarial networks can help with that because they support unsupervised and weakly supervised learning, which means we, in the unsupervised way, we don't need any label, and in the weakly supervised, we just need the label that this image has cancer in it. GANs enable distribution learning, moves beyond traditional supervised learning of decision boundaries, works with limited data. In my second study, we used only 266 patients, as we can see, as we will see, and then can enhance data augmentation and give synthetic image generation. Overall, GANs have been applied in medical images for reconstruction, synthesis detection, segmentation, and denoising, and so on. However, despite the potential benefits, the application specifically of GANs for the detection and localization of PCA with MR images has not been sufficiently explored. Therefore, the aim of this study is to leverage this inherent efficiency of GANs using the unsupervised and weakly supervised learning approaches to improve detection and localization on prostate cancer on MR images. And then we want also to integrate these GANs in an automate end-to-end pipeline CAD system. Now let's see a summary of the paper and how those papers in my study connected together. So for the first paper, we have built a tool. We call it CROPro. And the main reason for that tool is to standardize the way that we're taking the area of the prostate to train the models. And this is specifically important for GANs because we don't want to give them a lot of noise when we're training them. Then next, both paper 2 and paper 3 makes use of paper 1. And then paper 2 is basically finding, benchmarking which GAN is the best candidate for those that are available to test it on the final detection localization of clinical significant prostate cancer. Here we used only T2 weighted just to prove that it can work, and we try to have any PCA as a detection. In the last paper, we took the best found model from the second paper, and then we used it on the biparametric modalities, all the sequences, to clinical significant PCA detection and localization. So now let's dip to the models, to the papers. We have the first paper, CROPro, a tool for automated cropping of prostate magnetic resonance images that helps in the pre-processing steps. And as I said before, this is important because we don't want to have the surrounding noise. We don't have to have a lot of surrounding noise. And here you have an example of the apex, middle, and the base. And what is interesting here is that the size of the prostate changes through those slices. So usually people will crop those images just in a bigger size to fit it. And there are a lot of different variations between scans, where there are studies actually, if they don't get the information of where the segmentation is, then they might assume that the prostate is in the middle of the picture, which means that if it's not, then they will crop it wrongly. So overall, we find that there was limited attention when it comes to input image sizes and how they impact the deep learning model performance. And there was a lack of standardized way of cropping these prostate areas, that there was a need to the automated CAD systems. So the aim of this study was to develop a robust, flexible, and accurate tool for doing so, and to provide a standardized approach for that, ensure it's capable with the any deep learning model, and to balance this information of prostate and surrounding. To do so, we used transfer learning. We took 5 CNN models, convolution neural network models, and 5 visual transformer models. I'm not going deep on those models, but the task that we did, it was on image level detection for the clinical significant prostate cancer classification. And the CROPro can automatically crop MR images regardless of patient condition, image size, prostate volume, and pixel spacing. And then we have used the center cropping as a baseline. And then we have introduced 2 sampling strategies, random and stride, where random is basically take the prostate area and then randomly chooses to crop prostate parts. And then where the stride starts from the top left and go through the whole prostate area. And then here is the cropping settings and then the pixel spacing that we have used. The dataset that we used was the PKI dataset, and here is the split. And by running these experiments, we found that by cropping more focused to the prostate, we could improve the overall model performance compared with the baseline, where it was 40 centimeters compared with 10. So overall, the performance on clinical significant PCA on image level classification of both of the CNNs and VITs, vision transformers, depended on the CROPic setting. And we demonstrated that the CROPro is well suited to optimize these settings in a standardized manner, which could improve our overall performance of deep learning models. Now that we have that, we move to the GAN methods, and I will start just explain a little bit again about GANs. So we have the GANs here, and we have the generator that takes an input noise and then tries to generate a fake image. And then we have the discriminator, which is a binary classification model, basically, that takes both of the training input and fake and then try to predict if it's real and fake. So now, how do we leverage that for this work was that we train those models to generate only negative images, only healthy images. So we give an original image to the model, and then that model can remove parts of the cancer area that believes exists. So when we have that, then we can take the original image, the generated image, and then we can subtract that. Now, if we would not do this pre-processing step before, that image will have a lot of noise around it. That's the first step. The second step is to add some smart way of post-processing, and then we can show to the radiologists the detection maps. So this is for both second and third paper, similar approach. So the second paper has a title, A Comparison of Generative AI GANs for Automated Prostate Cancer Detection on T2 Weighted Images. And we wanted to evaluate it and compare several GANs models for automated prostate cancer detection on T2 weighted images. And we wanted to identify the most promising GAN for prostate cancer detection and benchmark that one in end-to-end CAD pipeline. Now we searched in the literature, and then we found, first of all, interesting models and available. So here we have 6 of them. Two of them are unsupervised, and the rest are weakly supervised. And then we train that for PCA detection on patient level using the area under the curve. And the dataset was for training was an in-house dataset here from the hospital, and we used the Prostate X, by that time a good dataset for online available for testing as external dataset. Overall, the results showed that one of those models with weakly supervised has the best AUC. We picked that. We also visualized that. We visualized if the results make sense. So we had the cancer over there, and then we saw that the model, that specific one, can identify that. And then we took that one, and then we tested. We ran it 5 times, and then we tested on the internal and external test set. Overall, the results on validation, internal and external AUC are stable. And also, when we visualize that, we could see that the model finds a similar area as a cancer. However, this model is not perfect, and there are cases where it fails. Whether it's the model or the sequence itself is a question. So to conclude, for this paper, this work shows that GANs have potential for detection of any prostate cancer and serve a solid basis for further development, where we can take the best found model for detection of more clinical question that we're interested in, detection and localization of clinical significant prostate cancer on a larger dataset. And not just using the T2 weighted images, but the biparametric images. So the last paper is called Improving Prostate Cancer Detection and Localization Using Biparametric MRI. Here we want to use GANs for detection and localization of clinical significant prostate cancer on biparametric MRI. And our hypothesis was by including the biparametric MRI, the overall performance will be increased compared with the second paper. So the methods that we used, we took first the best found model from the second study. We used that as a baseline. And then we did search in the literature, and then we proposed a variant of that model, which use spectral normalization and regulation, R1 and R2, as proposed. And then we tested on the clinical significant prostate cancer detection and localization for patient levels. And we used the area under the curve, the average precision, and a combination of those two, which is the average of both of them. The dataset that we used for training was an online available dataset, the two of them, PI and Prostate 158, train validation and test. And then we used a strong dataset that we had here from the hospital with 3 years follow-up after the MRI. The way that we did it was that we trained 9 models overall, 3 for each sequence, and then we picked different iterations of that model that has a good AUCs on the validation set, and then we added some strategies where we merged in different ways those models. Overall, we found that the best approach was combining all the 9 models with the first top AUC in our results that we will shortly see. The pipeline is the overall cut pipeline, it's a bit more complicated, but it's an extension of paper 2 as well. So let's dip to the results now. We have the validation results on the baseline. You can see an AUC of 825 and average precision 52.66, and then the overall PI was 0.676. And then we tried that was the best from all the merging techniques that we did, and then we tried that for the proposed model, and then we found that the results were increased. We can see that from 0.67 went to 0.71, and also the AUC and other precision have been increased, and that was also also with the other, in combination with the other sampling techniques. And then we had that as first results, and then we took that model and then we tested on the internal and external dataset, where in the internal we had a PI score of 0.57, and the external we had a result of 0.75. Now, another example. Here is just an example of how the results look like. We have the original images. We have the GAN detection maps. This is in 3D. This is just a slide of it. And then here we have the human lesion labels. In conclusion, an automated cut pipeline containing GANs trained on biparametric MRI has successfully used to detect and localize clinical significant PCA. The proposed GAN improved the model performance compared with the baseline that we had. And then the sampling technique, those merging different models, states of the models, has shown to improve the overall performance of the model. So now for the discussion, advantages on deep learning based CAD system can overcome the limitations in traditional diagnostic approaches. The GANs have shown potential for clinical significant prostate cancer, both detection and localization, which can reduce the need on these detailed annotations. And there is challenges in clinical AI adoption, where there might be variations across different scanners, datasets, and institutions. So let's now see some of the limitations and the future work. So some of the limitations can be that there is variability among MRI scanners. GANs can have problems with stability, training instabilities. We don't have universal evaluation metrics for those, and in general, explainability is a challenge. Overall, there are alternative generative models, there are plenty of them, and improvements in both explainability and trust in AI as a necessity, and the integration into those in the clinical workflow is needed. In conclusion, I want to say that this thesis makes an important contribution to the field by demonstrating the successful integration of GANs into CAD systems for PCA detection and localization. And this work lays as a foundation for future research aimed to overcoming the traditional limitation in PCA diagnosis, especially those related to the lack of annotated data. And with that, I would like to thank you for your attention.